Okay. Well, good afternoon, everyone. We're going to begin. My name is Chris Krahansel, and I have the privilege of overseeing the business development and the client engagement efforts here at Biologics Consulting. Thank you very much for joining our event today. And our discussion is part one in our combination products series. For today, our presenters will talk about the integration of drugs and delivery devices, and we are grateful that you came and that you took time to tune in. There will be some time at the end of today's presentation for some Q&A, and at the top of your screen, let me go back here, letting more people in. At the top of your screen, you can use the Q&A button to ask your questions to any of the panelists, and you are strongly encouraged to reach out to today's presenters if you have any additional questions after our time is over. Here at Biologics Consulting, we do what we do for the good of public health, and we want to be able to share that information with you as well. So thank you again for joining us today. And so without further delay, allow me to introduce our presenters. First, we have Anchal Chowdhury, Senior Consultant for Medical Device, and she's a Senior Consultant here at Biologics Consulting. And also Christina Vesely, Senior Consultant for the CMC team here at Biologics Consulting. And so I'm going to pass it over to Anchal. And so once again, thank you very much for joining us today.
Thank you, Chris. This is our first webinar in our combination product series that focuses on the integration of drugs and devices from a pharmaceutical and biotech company perspective. So we'll cover what these companies need to know and think about for delivery device development with a focus on the U.S. market. And just to note, we'll be using the terms drug and drug delivery, but most of the same information does apply to the delivery of biologics as well. So I first wanted to just start off on the why of delivery device development. Why choose to integrate delivery devices into your program? So these devices allow for more targeted drug delivery, a lot of times improved dosing accuracy, user convenience. Sometimes that also comes along with improved adherence, a reduction in the contamination potential, and long-term cost savings by cutting down on wasted product. Because of these reasons, the drug delivery combination product market has really been continuing to expand over the past few years. So drug delivery devices range a lot in complexity. They can be single-use disposables like pre-filled syringes, all the way to implantable devices like intrathecal infusion pumps. And the route of administration can also vary from oral or nasal to subcutaneous or transdermal or vaginal. But when any of these devices are marketed to deliver a specific drug, they are considered to be combination products. So FDA defines a combination product as a product composed of any combination of a drug and a device, a biologic and a device, a drug and a biologic, or a drug, device, and a biologic. And we're focusing today on just the first two categories of a drug and device, or biologic and device. So combination products can be configured as either single entity devices, like single entity products like a pre-filled syringe, co-packaged where the drug and the device are shipped together, but they still need to be assembled at the point of use. They could also be cross-labeled where the drug and device parts are shipped and provided separately, but they're labeled to be used only with each other. And finally, even at the investigational stage, cross-labeled products are considered combination products. So once you've determined that you want to pursue adding a delivery device to your drug program, you'll want to think about the development process and also the regulatory approval pathway. So starting with kind of the end in mind of regulatory approval, in the US, the regulatory pathway will depend on the combination product's primary mode of action. And this is the mode that it provides the most important therapeutic action. And oftentimes this is the drug or the biologic, but that's not always the case. Sometimes it's the device that provides the PMOA. So if you're not sure about what the primary mode of action for your product is, we can help with that. Sometimes a request or a pre-request for designation is needed to get an opinion from FDA on which center will lead the review of the product. But once that's decided, who's the lead center is decided, you can approach the appropriate center for product-specific meetings related to development. So these, for CDER and CBER, these are type A, B, C, and D meetings. For CDRH, they can be pre-submission meetings, submission issue requests, or for PMA's day 100 meetings. And relatively recently, FDA has also added combination product agreement meetings as an option for combination product sponsors. So now kind of to get back to the beginning of development, let's talk about the device constituent part regulatory considerations. An important part, one of the first things you probably want to do is ensuring that you have an effective quality management system in place that meets the CGMP requirements. So if you're a drug or a pharma or biotech company developing a co-packaged or single entity combo product, and you already have procedures in place to follow parts 210 and 211, then you only need to meet the additional subset of device quality requirements that are outlined here. Typically, installation and servicing don't apply, but it is very important to have these other pieces like management responsibility, design controls, and purchasing controls and CAPAs in place. So, even if you are using a device partner, it is the sponsor's responsibility to have these pieces in place. So I will say that design controls are kind of the main focus for device development, and we'll go through that in the next slides. But I did also want to mention that FDA has issued a final ruling updating the 820 regulation to the quality management system regulation, and this will go into effect in February of 2026. This update is not going to change the fact that management responsibility, design controls, and purchasing controls and CAPAs are needed, but it will change the 820 regulation to better align with ISO 13485, and it'll more explicitly spell out what is expected. So if you are looking to establish a device quality system, this is something to keep in mind. So talking about the design controls, this is kind of the heart of device development. It's based on a design control framework to help ensure quality. Establishing design controls includes determining the user risks, the design user needs and inputs, the outputs, designing the manufacturing process and supplier qualifications to build the device, and then doing verification and validation testing. And what's not shown here is the design transfer and then the commercialization piece. And this can all be a very long process, and standalone device development takes typically between 3 to 7 years. So pharma or biotech companies who are developing their own delivery devices should start that device design process and testing early. You'll want to at least have a first iteration kind of a basic device to be used in early clinical studies. The design and build and verification validation steps will likely need to be repeated between clinical and commercial versions of the device when there's significant changes, but it's good to kind of start the process early.
And Ansha, if you don't mind, I wanted to jump in with a question. A lot of the drug companies that I work with aren't really implementing their devices until phase 3, especially for an already marketed device like a prefilled syringe. Is that a problem? I mean, can you comment on that at all?
Yeah, thank you, Chris. So you're right. So for devices that are already marketed, they can be introduced in phase 3. The recommendation is for all devices, whether they be novel or already on the market, that the kind of final version of the device be integrated and tested during phase 3 clinical studies. So if a company is using an off-the-shelf already marketed device, they should be able to add it during phase 3 without any problems, as long as they take into consideration that it's the same indication and delivery, route of administration, things like that.
Okay, great. Thank you.
Mm-hmm. But sometimes companies do run into situations where the final device design changes kind of significantly partway through phase 3, or even after you've completed phase 3 studies. And in these cases, it can be really beneficial to get expert assistance on a bridging strategy or talk, and also talk to regulatory agencies. So other device development considerations. So while you'll need that QMS aspect for any drug delivery combo product, there are also device-specific considerations that are listed here. Not all of these may be applicable to your specific delivery device. But many of the items listed, like biocompatibility, sterility, electrical safety, EMC, they're considered kind of essential for safety, to establish device safety. So FDA would expect that those be fully assessed prior to introducing a device into an IND. Performance aspects like human factors, manufacturing, this can be kind of introduced later on. So in terms of the delivery, the performance of the delivery device, FDA gives special consideration to what is now being called essential drug delivery outputs, or EDDOs. Draft guidance on this topic was just released on July 1st. And FDA has defined EDDOs as the design outputs necessary to ensure appropriate drug delivery. And these are outputs associated with dose delivery, delivery to the target site, production preparation, initiation, progression, and completion of the dose delivery. And these are kind of analogous to CQAs in the drug world.
And I wanted to jump in again. I know that you're talking primarily about novel devices, but does this new guidance apply also to those off-the-shelf components like the prefilled syringes and things that are already marketed?
Actually, yeah, it does. So these EDDOs should be considered for any device, even if you're using something that's on the market. As part of that kind of design control process, you need to look at what are these essential drug delivery outputs, and are they, if they haven't been appropriately verified and validated by the CMO, then that needs to be done, and it definitely needs to be done in combination with the specific drug product that is, or the biologic that's going to be used.
Okay, thank you.
So yeah, FDA does expect the V&V testing and manufacturing controls to be identified for these, is what they are terming EDDOs, or essential design outputs. And just to clarify, it's not just on that V&V testing is needed for those essential outputs, but it's needed to address all design inputs and outputs. It's just that there's certain special focus given to EDDOs where the testing is expected to be done in more kind of worst case conditions. So preconditioning is expected. It's expected that that when you're testing these specific design outputs, they've already undergone shipping and transport type testing, and testing should be done at end of life. Additionally, as I mentioned, there needs to be the manufacturing control strategy, or these need to be tested at batch release to kind of really verify that every lot of product is performing properly. It's also important that the clinical studies that are being conducted have some endpoints that address the drug delivery performance. So now getting to if you are using a marketed device or off-the-shelf device, a lot of the work will be done, but not all of it. You will still need to determine if the device is being used according to its already marketed indication, route of administration. And if not, then you will want to probably integrate its use earlier on in clinical development. Otherwise, like we mentioned, it would be fine to bring it in during phase 3. You'll also need to set up a quality agreement with the device manufacturer to talk, yeah, Chris, go ahead.
Sorry, go ahead. I'll let you finish and then I'll ask my question. I was just going to mention that in addition to the device manufacturer who actually makes whatever this component is, we also need to make sure we have quality agreements in place with whomever is helping us integrate our drug into that device. So thinking about a fill finish process, we would have to have a fill finish vendor to actually work with to get the drug into like a pre-filled syringe or other types of devices as well.
Yeah, thank you. That's a great point. And then kind of more on the regulatory approval side, you'll want to have a letter of authorization from your device manufacturer so that FDA can reference their submission while also reviewing yours. And you should also be prepared to conduct additional studies on the safety and performance of the device when it's used with your drug or biologic. And with that said, I'm going to hand it over to Chris Vesely to discuss the next few slides.
Sure, thanks. And we can jump right into the drug constituent part. So the first aspect, of course, is to make sure that we have control of our drug product. And when we're talking about a combination product, we need to not just be looking at the functionality aspects, but we need to have a specification that looks at all of our expected testing to assure safety, identity, strength, purity, and quality. So that is going to include everything that we would normally have on our specification. All parenteral testing requirements must also be met for injectable products, even if we're working with some sort of a device. So that includes parameters around color and clarity, the presence of visible or subvisible particulates, endotoxin, sterility, container closure, integrity testing. But one thing to note is that some of these devices really create challenges with performing some of the more routine tests, particularly things like visual inspection of the product to confirm appearance. When you have something that's now inside of a device, it may be difficult to actually view that product within the device to confirm it's still meeting those criteria. And then there are other things that come along with devices. For example, a lot of our prefilled syringes are siliconized. Some of these processes that we use to make sure our devices are going to be functional will impact certain aspects of product quality. we may see increases in subvisible particulate matter that may or may not be relevant to any changes with the quality of the drug product. And so we need to be aware of that, and we need to make sure that we have testing that is set up that will enable us to fully evaluate that the product is of suitable quality for human use. The other thing to note is that we don't want to look at this just at the time that we're releasing, but we need to be evaluating this across shelf life. So one thing to note is that there's two different ways to look at this. When we're talking about product performance, we want to think about how we're storing this. What is our long-term storage condition? Is it just the product that's filled into a syringe or into a cartridge? Or is it the product after it's been assembled into some other device, such as an auto-injector? And using the examples here, you may have a product that's filled into cartridges that is immediately assembled into that auto-injector, or it may be stored as cartridges and then only supplied into the auto-injector at the time of patient use. In the United States, again, the FDA expects we're going to look at the long-term storage for however we're really going to store that in the long term. And it's important that if you have your product already inserted in that device, that you have that as your stability configuration. Whereas in Europe, there's a little more tolerance to looking at your stability more in that primary container closure system. So again, thinking about the cartridge as opposed to necessarily having to have that device on stability. So there certainly are some differences from region to region, and we can help you figure out exactly how you need to have your product configured for those long-term stability studies. But just another further complication to consider as you're setting things up. Moving into kind of the general parameters around integration of your product into a device. The first aspect is component selection. We need to make sure that we're selecting components that are going to be reasonably compatible with the device and with the drug. So some of the key aspects we need to think about as we're considering what those components are going to be. What is the drug category? Are we dealing with a small molecule? Is this a protein or other type of therapy? What is the formulation? Is it anything unusual in terms of pH or excipients? Do we have any known incompatibilities to certain types of plastics or other components that are used commonly in devices? What volume or concentration are we going to be dosing? You can imagine that the device selection is going to be significantly impacted based on whether we need to deliver a couple of mils versus 20 mils of solution. What is our route of administration? Are we looking at something that's IV? Is it subcutaneous? Is there some other mechanism by which we're going to administer the product? And how much time do we need for the administration? If you think about the example of a prefilled syringe versus an on-body device that's going to deliver over a couple of days, that's going to have a potentially significant impact on product stability. So we need to make sure that as we're selecting the components, we're taking that into consideration. We're thinking about what temperature the drug is going to be exposed to through the duration of the injection. We also need to be thinking about where this is going to be administered. Is this something that is going to happen only in a doctor's office? Or is the patient going to actually take this home and be responsible for their own administration? Because that's going to impact how we're going to select our components as well. And then the last point I wanted to make is that as we're selecting our components, we also, sorry, if you can go back one slide, sorry. We want to make sure that we are considering the quality of components. And we know from our experience over the last couple of years that going back to the COVID times, there was a significant issue with availability of certain components. And so as we're selecting our components, even today, we need to make sure that we have adequate supply, that we have the availability of the components that we need. We need to make sure that we have good partners to work with, whether that's for the components themselves or those groups that are manufacturing things for us, and we need to make sure that we have good relationships and adequate capacity to fill our needs. And now moving on to material compatibility studies. Material compatibility sounds like a simple topic, and it really is, but what we're trying to do here is we're trying to demonstrate that we have a lack of interaction of our drug product with the device itself, and that device is not going to adversely impact the quality of our drug product. The agency expectations are that whatever studies you're doing to assure your material compatibility, that they are going to be representative of the actual use and/or storage conditions for the product. The obvious thing we need to think about is the chemical and physical compatibility of our drug product with the device. So things we're looking for are, do we have new or different impurities that are forming when we have this product in the device? Are they forming at an increased rate? Do we have other events occurring, such as protein aggregation due to the presence of silicon oil or some components of our device? Another big one is, are we losing our drug product to the device surface? If that's the case, then that's going to also impact the dosing to the patients. In addition to thinking through the long-term stability for the chemical and biophysical product itself, we need to think about how the device performance may also be impacted by shelf life. And you can imagine that some of these devices are very finely tuned in terms of how they're dosed and how they're how the device component is set up. And if we have minor changes to the product quality on the drug side, for example, slight increases in viscosity with storage, that can actually impact our device performance, which can impact your ultimate dosing to the patient. So we need to be following that across shelf life as part of our material compatibility studies. And then another very big topic that I'm only going to hit the surface of is extractables and leachables testing. Excuse me, just for one moment. So the extractables and leachables testing is required, especially when we have components that are including plastics, metals, and other things that can leach into our product over time. And so the first component of this is actually the extractable testing to determine what are the potential items that could extract into our product or leach into our product over time. We also need to define our analytical evaluation threshold before we start into our leachable studies. This is a value that tells us at what point we need to be concerned if we have a minor peak that shows up in these studies. And then we need to think about how we're going to perform a safety evaluation of anything we see increasing across shelf life. And then in special cases where we do see peaks that are of concern, that are growing over time, we need to think about mitigation. And that may mean choosing different components, going back to that first step of component selection to choose either plastics that are more compatible with our product, or maybe different levels of siliconization, different qualities of components that may impact the overall extractables and leachables profiles.
Thanks, Chris. So I'm just going to talk about the common pitfalls when it comes to integrating drugs and devices. So one of the biggest traps that I've seen pharma and biotech companies falling into is siloing device and drug development. So yes, you definitely do want a separate device team who is experienced with the device development, but there really needs to be really good communication between the teams and a strategy from the start to align and integrate development of the two parts. So you don't end up in a situation where the drug is ready to go, but there are still issues with the device, or vice versa. Another issue is overdependence on a device partner. So if you are leveraging a master file from another company or marketing application, there may be items related to your marketing application that the device partner will need to respond to. However, you are, as the pharma or biotech company, you're still going to have an understanding of what those issues are, the timeline the device partner has to address them. And I've seen this as an area where consultants like us can help both the pharma or biotech companies and the device company addressing FDA's or other regulatory bodies' concerns, while kind of keeping each entity's IP safe from the other. So that's something to think about if you are going down the pathway of using a master file for another device company. Other pitfalls include not considering those essential performance or EDDOs, what's now called essential drug delivery outputs, early in the combination product development. You definitely want to identify those early, since they need to be tested at kind of those worst case conditions and at the end of shelf life. Also, not planning device manufacturing controls along at the same time that you're looking at the CMC drug controls. That can kind of set you back once you're ready to actually commercialize. And then also not considering the need for human factor studies. It's something that pharma companies are not usually experienced with. I've seen that be kind of a hurdle for them. And then I'll just add to this what Chris was mentioning about extractables and leachables testing. Those tests can take a long time, and a lot of times FDA likes to see them done in a very specific way, so it can be helpful to get some input from them on those ENL testing protocols before conducting those tests. So just to summarize what we talked about, combination products definitely offer advantages for patients and even for pharma and biotech companies. FDA regulates combination products based on their primary mode of action. The device development piece does require implementing design controls in parallel with a risk management process. There are some unique challenges when partnering with another firm to develop a combo product. And then the drug product testing can be more challenging than testing a drug product on its own. It's very important to consider those drug and device interactions. And I think that is all we have. I'll move on to questions and Q&A if anyone has any questions from the audience.
And I would add, we'd love to have this as a conversation, so please feel free to jump in with questions. We can have a dialogue or debate if needed.
And also, Ted, we're going to be having a few more webinars on combination products. So our next one will probably be focused on device companies looking to enter the pharma market, and then talking about some hot topics like digital health and human factors. So
we have a question. I don't know, Angela and Chris, can you see the question in the QA area?
Yes.
See.
If you can, I can read it, but I wanted to see if you see it there from Daniel.
Yes, we can see it. So this is a question about whether a topical biologic being delivered by a plastic pre-filled syringe without a needle needs to follow the new EDDOs and have them be established prior to phase 1. Since this is an off-the-shelf device. Anshul, do you want to take this one?
Yeah, I would say probably not prior to phase 1, but if it's off the shelf, then around phase 2 or phase 3, it's something that you would want to think about, and definitely before phase 3, test those to make sure that with your drug product, those essential design outputs are being met satisfactorily.
And it looks like there's also a human factors question on that at the very bottom.
Yeah, human factors definitely do not need to be conducted prior to phase 1. They can actually be typical. So you want to do some initial usability testing in phase 2, and a formative testing during phase 2, and then the actual summative testing can be done in parallel with phase 3.
Thank you very much. That was very helpful.
Mm-hmm.
Okay, and I will go ahead and address the next question, which is a chemical and biophysical compatibility question. So a lot of the products that we work with don't necessarily have a like a USP reference standard. Most of our drug products have some sort of internal reference standard that we're working with. And so our criteria that we're going to use for assessing chemical and biophysical compatibility, we're going to start with that release panel for the drug product. But we're going to include extended characterization to look for things like, if we're talking about a protein, we're going to look for aggregation or higher order aggregate formation, such as particulate formation. We may be assessing other biophysical properties, such as primary, secondary, tertiary structure. As far as how we're going to relate this to our extractables and leachables data, that's a very complicated question. A lot of our assays that we're using in the laboratory to look at drug product quality may not see anything with respect to those components that may be extracted from our product, but we may see a change in stability of the product. So a lot of this comes down to looking at the long-term stability of that API in the presence of the drug product device versus just in a glass vial. So just doing that comparative analysis between the two systems. If you have certain extractables, you may look at specific extractable studies or leachable studies where you're actually spiking that into your drug product to make sure that there's no adverse interaction, especially in the case where you're trying to get an early read on this, rather than waiting until you're 2 years into a shelf life study to determine if there's an interaction or a problem.
And then I see a question, says often clients ask for the most expedited pathway using an existing device or to use an existing device or develop a unique device. What are the pros and cons, and what are the hurdles and timelines for development of a unique device? So, using an existing device definitely does cut back on some of the activities. But like I mentioned, there's still several things that a pharma company or biotech company is going to have to do to ensure that drug device integration, that like material compatibility that Chris was talking about, to ensure that the performance, that the device is performing appropriately with the drug. So it does cut back a bit on the time, but there's still some testing that needs to be done, and then that adds that additional complexity of working with another partner. The timelines for standalone device development, I mentioned, is usually between 3 to 7 years. If you're taking something off the shelf, already marketed, and you can usually, most of the time, not all the time, but a lot of times introduce that in phase 3. So you're looking at probably saving maybe a year or two on, or a couple of years at least, on the design and some of that early phase development. So it really just kind of depends on the unique application, what the company feels from a business perspective might be best for them. There's a lot of different considerations there.
And I would add to that that the limitation a lot of times with the off-the-shelf components and already marketed devices is that they may not be suitable for your particular application. If you're looking to deliver 20 mils of solution to a very specific location, there may not be a device that's easily available to you. So you may be in a situation where you have to either partner with someone who's still working on that, or you may need to develop your own. So that's something to consider as well.
We also have a question that came through the chat. Does all the testing you mentioned for the device need to be completed for an IND?
Yeah, it's a good question. So a lot of the safety related things do. So that's why I was mentioning like biocompatibility, sterility, electrical safety, if that's applicable. Those type of testing would need to be completed prior to an IND. The kind of that EDDO testing, I think FDA hasn't really spoken that clearly in this draft guidance on whether that needs to be done at the IND stage. In my experience, you want to identify what your EDDOs are, but you may not need to do them at, for example, end of life at the time of IND, because you probably don't even know what your end of life is yet. So it kind of, you have to take each test and see, does it make sense to do it at this point or not?
And I think it's in your best interest also to at least perform the basic material compatibility studies, because you don't really want to start into even a phase 1 IND if you have something that's completely incompatible with the drug that's going to ultimately make the drug either not safe or not efficacious, because that's only going to set you further behind.
Yeah, so that's a good point. So extractables and leachables, usually a leachable study is done prior to an initial IND, opening IND, just to make sure that there's no that there's no real safety concerns, and then kind of the more narrow extractables testing would be done a little bit later on.
Okay.
And thank you for that. Just to follow up on that, what about some of the stability studies that you mentioned? Do those need to be also? I mean, you have to have some shelf life expectation, obviously, before you submit. But how does that play into all of this?
Sure, and that's a really good question. So stability is always a little bit tricky. It depends on exactly how you're configuring that device, but most of the time for a phase 1 IND, the expectation is that you're going to have at least 3 months of shelf life in something that is representative of what is going to actually be used with patients. And that's kind of your minimum requirement most of the time. And what lot that is going to depend a little bit on how complex is your system, how off the shelf or how unique is the device that you're working with. If you're working with something that's just a pre-filled syringe and you've already had your product in glass vials stored for some amount of time, that's a pretty easy switch. But if you're going to something that's more like an on-body device where it's going to be in direct contact with a number of plastics for a very long time, even during storage, then that would be a different situation. And I mean, we can certainly talk in more detail if you're interested about recommendations, but the agencies are going to expect at least some stability data going into that phase 1 IND.
Thank you.
Well, we're at the end of our time here, and I just want to say thank you again to Anshul and also for Christina for the information. we had one more question here. Before I even closed out, we got one more here that came through the chat. We can take one more and then we'll have to close. For combination products, where there is a supplied diluent, do you have any comments, suggestions about that diluent or reconstitution only being filled with a biologic?
I do have a lot of opinions on the topic, but it's really going to come down to what is your biologic and what is compatible with it. The obvious diluents that are out there are saline, there's dextrose, those are off-the-shelf components, but even those are going to have potential compatibility issues depending on the exact product you're working with. If it's supplied with a diluent and it's supplied by that drug manufacturer, it's probably best to work with that because that's already established as being
Mhm.
compatible with the drug itself. But we can certainly again talk about that in more detail and provide recommendations if we had more detail on the product type.
Great. Thank you. Thank you for the question. Thank you very much for participating in today's webinar. Again, Anshul, Christina, thank you very much for the wonderful information. And as you can all see on the screen, we will have some upcoming webinars to continue with our combination product series. So thank you once again from Biologics Consulting. I'm Chris Krahansel, and it's a pleasure having you join us today, and we'll see you next time. Take care, everybody. Bye-bye.
Thanks.
Thank you.